Good morning, students. Today we will have to discuss the embryonic development in human being. This is one of the part of the reproduction. Before parturition, what happens in zygote? As you know that the zygote formation takes place in the fallopian tube, and after 30 hours of the fertilization, before 30 hours of the fertilization, the zygote is still there, having an outer covering that is called zona pellucida, and no changes, a very slight changes will be takes place. But after 30th hours, what happens? The cell divided into two unequal parts, and that is called first cleavage. That is called first cleavage. This is the product. The slightly shape is slightly different. One is small, another is larger. Gradually it has been observed in the 40th hours, it is 4-celled stage, and in third day there is 8 to 16 cell stage. Now, in fourth day, this 8 to 16 ball-like structure, blastomere-like structure, that is called, this is the solid, this type of structure, solid mass of cells, it is called morula, because externally it looks like a spotted structure, so it is called morula, and having a well developed zona pellucida is still present on it, and that will be shifted into the uterus. There, inside uterus, they absorb uterine fluid, and this particular 16 to 32 cell stage is differentiated, or the shifting of the cells will form this type of a structure, and that is called blastula. It is called blastula. What happens from morula to blastula we will discuss. Blastula. This is blastula because it has a blastocele, and it has been observed that the cells has been shifted like this. It is called peripheral cell, that is called trophoblast or trophectoderm. Trophoblast or trophectoderm. And this part, the certain cells are shifted in the apical part, that is called animal pole, and this is called inert cell mass. Inert cell mass. The trophectoderm of this particular region, this trophectoderms is called cells of Robber. Actually, this inner cell mass is the embryonic part of this structure. Here embryo is formed, and this trophoblast or the trophectoderm will form extra embryonic membrane, that is extra embryonic ectoderm, mesoderm, and we will discuss it, how this will convert into ectoderm, endoderm and mesoderm, and how this will convert it into the embryos. In this figure we will see that it is the uterine epithelium, means endometrial lining is just come in contact with this one. And whenever this proper place will be gained by this particular embryo, then what happens? Gradually this zona pellucida disappeared. Now there is no function of this zona pellucida. The function of this zona pellucida to select the proper site for the plantation. Okay. Now it will have to completely insert inside the endometrial lining or the uterine epithelium. So for this purposes, what happens? They will form two type of cells. One is the cytotrophoblast and another is the syncytotrophoblast. This syncytotrophoblast will help this particular to insert inside it, and gradually what happens? They cover like this. Now this whole of the embryo, this whole of the blastula is settled down inside the endometrial lining or the uterine epithelium, and here they form the placenta. But before formation of the placenta, what changes will take place, just we will have to discuss. Now first of all, we will see that this is the trophoblast. This is the inner cell mass. Inner cell mass. This one is the cell of Robber, and now it is the zona pellucida is absent. Now what happens? There is change in this particular inner cell mass. What changes? Just we have discussed. Here we will see that this trophoblast is still here. And this inner cell mass is divided into two type of cells. One is called epiblast and another is called hypoblast. Gradually, this epiblast and hypoblast shifted towards the blastocele, and now it is called primary yolk sac. Primary yolk sac. Now what is the next change in this particular? We will discuss here that what happens. It is still like this. It is the epiblast, this one is the hypoblast, and it has been observed this cell of Robber will secrete a layer of the cells. It is called amniogenic cell. It is called amniogenic layer or amniogenic cells. That is from the trophoblast or the cell of the Robber. And this particular cavity which is in between the amniogenic layer and the epiblast is called amniogenic cavity या amniotic cavity. Amniotic cavity. Now this hypoblast, suppose this one is the hypoblast. form a layer and this is also called the same thing, primary yolk sac, and this layer is called endodermal cell. This layer is called endodermal cell. Now, what will be the next development? In this embryo, just we have seen here that the embryonic endoderm is being formed and which is just above the trophoblast. What will be the next change? Just we have discussed here. Now, there is no change in the upper part or the vaginal pole. It is still here. There is no change. It is the cell of Robert. This is the amniotic layer, and this will is the amniotic cavity, and this is the epiblast. And this endoderms are also being developed to form the primary yolk sac. And what is the next change, second change, just we have discussed. Now it has been observed that from trophectoblast proliferation will be started. And this type of cells is being formed from the trophectoblast. And this is called extra embryonic mesoderm. Now we'll see here that the extra embryonic endoderm and extra embryonic mesoderm is being formed, and now still it is called primary yolk sac and this is called amniotic cavity. Now what happens, there is a certain change in this particular extra embryonic mesoderm. It means shifting of this extra embryonic mesoderm. What types? Just see here that this is the trophoblast, this one is the hypoblast, this one is the epiblast, this one is the amniotic layer, this one is the amniotic cavity, this is the extra embryonic endoderm, and it has been observed that this extra embryonic mesoderm, which is still present here, will be shifted like this. It means a cavity is being developed. And this is called extra embryonic coelom. The outer extra mesodermal part is called somatopleuric extra embryonic mesoderm, and internal mesoderm is called splanchnopluric extra embryonic mesoderm. That is called somatopleuric extra embryonic mesoderm, and that is called splanchnopleuric extra embryonic mesoderm. Now it is called secondary yolk sac. Secondary yolk sac. Here it is the time of the formation of this particular epiblast and hypoblast into the primitive form of the embryo to form the ectoderm, mesoderm and endoderm. How this will develop into the embryo? Actually it is the embryonal lobe, so they are responsible to form the embryo. Previously we have told you, and this is responsible for the trophoblast is responsible for the formation of the extra embryonic membrane. Just we have observed here that the extra embryonic endoderm, extra embryonic mesoderm, and this mesoderm has been divided into two parts, that is the splanchnic mesoderm and the somatopleuric mesoderm, and with the help of this particular division it has been divided into extra embryonic coelom, that is called secondary yolk sac, and the amniotic cavity. Now we will have to discuss that how chorionic layer, embryonic layer, amniotic layer, chorionic layer, yolk sac are developed, and how this embryo will form the three germ layers. Now we'll have to discuss that how primitive stick is being formed. Now what happens? We will see that this type of structure. This type of structure has been developed, and this is the endoderm, and this whole the complete structure is called primitive stick. This complete is called primitive stick. It is the endoderm, and it has been observed that the mesoderms are developed in between this ectoderm and endoderm, and after formation of this one, it has been observed that the different tissues and organs are start to form. They are ectodermal in origin, endodermal in origin, and the mesodermal in origin, and now it is called triploblastic in nature. This part, and this part, this is called chorion. Here you will see that it includes trophoblast and somatopleuric extra embryonic mesoderm, and they are responsible, this part is responsible for the formation of the placenta. So the human being, it is called chorionic placenta, and this is called chorion. This part having trophoblast or the amniotic layer and mesoderm, that is called amnion, chorion, and this one is the yolk sac, like that. In this way, the placenta is developed. The placenta attached with the endodermal lining and embryonic lining, and nourishment will be continued, and the embryo will be developed till 9 plus minus 7 days. Whenever this phase is completed, the phase is completed, there is a special reflex that is called fetal ejection reflex. It is responsible for release of the oxytocin, which is from anterior lobe of the pituitary, and it is also called birth hormone. And they are responsible for the contraction in the uterus, that is called by the milking action, and there is a pressure in the vaginal zone for the ejection of the baby. Now at the meantime, relaxin is also being released, and they are responsible for the providing flexibility in the pubic symphysis for the normal birth. And it has been observed that the gradual increasing the concentration of the oxytocin is responsible for the pressure on the baby by contraction, and it is very much energy consuming as well as painful, so it is called labor pain. And after sometimes it has been observed during 2 or 3 phases, it is a long process, about some hours taking, and the baby will come out having this type of structure. Suppose this was the baby and this is called umbilical cord. This umbilical cord connected with the uterine wall with the baby. This is all about the parturition. Now one thing, that the baby is male or female. It can be detected at the time of fertilization, and that is called sex determination. That is called sex determination. It has been observed that a human being have 46 chromosomes in each and every cell, and out of that 44 is the autosome and the 2 are the sex chromosome. At the time of the gametization, what happens? 22 plus 1 and 22 plus 1 will be divided. Now this sex chromosome is of 2 types. One is the XX and one is the XY. When it is female, then their product is 22 plus X and 22 plus X. And when it is male, then their product is 22 plus Y and 22 plus X. This type of division, so the female is called homogametic and male is called heterogametic. Now, we know that the one gamete fused with the one gamete. If the gamete fused like this, then 44 plus XX, and if gamete fused like this, then 44 plus XY. In this condition, the baby is male, and this condition, baby is female. After the birth of the baby, it has been observed, just we have discussed previously that when a female will gain a particular age, when the FSH started, then estrogen, progesterone will be started, and the mammary gland, size of the mammary gland is also started. But whenever the female is pregnant and it is the time of the birth of baby, then a prolactin hormone is being released. And this prolactin is responsible for the increasing the milk retaining gland or the mammary gland. But at the time of birth or after the 24 hours of the birth, the milk will be started to secrete. And before that, that is called colostrum. Of course, the mammary gland become enlarged, the breast will be enlarged, and there is a liquid, a faint liquid is stored inside it, which is not actually milk, that is the pro-milk, and that is colostrum, which has a very good quantity of the antibodies. So it is very much essential for the baby to retain their immune capacity, to increase their immune capacity. So the milk, mother milk, is very much essential for the baby, because on that time the baby's immune system is not so much functional, and if they are taking their mother milk in form of the colostrum, their intensity of the immune will be increased and the baby become more survivable. That is whole about the embryonic development, parturition as well as the lactation, and this is the completion of the chapter.